Great.
Chat should be open to everyone. It is, so everyone be careful about what you type and to whom. I don't want Chris to have all the bad jokes. I'm gonna get a few in before during the intro.
Thanks.
Yeah.
Phones to silence.
Folks are joining. Lots of folks filtering in. Chris, if you want to go ahead and pull up slides on your end, I mean, I guess I could also just do it on my end, but
Yeah, I'll do it.
yeah, smoother that way.
At least I'll try to do it.
I believe in you. Welcome, folks who are joining us. We're just gonna let people kind of filter in for another minute here, maybe. I know timing's tight, so I'll probably not wait that long and just jump into housekeeping stuff, and then folks can sort of filter in as they can. You'll see the Menti link up there in the corner. There's some polls later on in the webinar. We recommend it in the interest of time if you want to pull that up on your device so you're just kind of ready to go when those polls actually launch. Might be a good idea while we're getting started. All right, I see we're up to pretty solid double digits, and we're a minute in, so I'm going to go ahead and launch us in. So welcome, everybody, to the Collaborative Science for Estuary series. This is Nick Sobral with the Science Collaborative. Today's webinar is a big multi-reserve collaborative research project. Specifically, it's a national study of marsh plant communities. I'm going to leave it at that level of detail for now because the speakers have way more knowledge and expertise than I could possibly bring to this, so I'm just going to leave it to them to talk through it. You'll hear more from them in a moment, but before we get started, on the next slide here, we've got a quick introduction to the reserve system, the Science Collaborative, and this series. So the National Estuarine Research Reserve System is a national network of 30 unique reserves as shown on this map. This is a NOAA program that works in collaboration with a local place-based partner, either a state agency, university, or nonprofit at each site. Each reserve site includes programs focused on the activities you see on each slide here, which is land stewardship, research and scientific monitoring, training programs to the public and local officials, and education. And the Science Collaborative supports science for estuary and coastal decision makers by coordinating regular funding opportunities and supporting user-driven collaborative research and transfer activities that address critical coastal management needs identified by the reserves. The Collaborative Science for Estuaries webinar series features project teams supported by the Science Collaborative, program staff, and partners. Speakers share their unique approaches to addressing current coastal and estuary management issues. And if that sounds interesting to you, please join us for future webinars to learn more about new methods to integrate experts and users into the research process and how the research results and products might inform your work. So on the next slide, we'll talk briefly about some program learning stuff that applies to webinars. Webinars are a valuable learning opportunity for our program. Insights from speakers and participants like yourselves can help inform how we support others in doing collaborative science, for example, in the development of future workshops, webinars, or resources such as tools, tips, and case studies to add to our guide to collaborative science, which is open access. With that in mind, we want to inform attendees that insights or feedback that you share, for example, in chat or via a survey or poll question, may be used for program learning and development. Please note that if we capture a direct quote from you that we like to use for learning purposes, we will reach out to you to seek permission first and provide an explanation of its use. All quotes would be used anonymously. We're grateful for the opportunity to learn alongside you. So lastly, last slide here, we've got some notes on housekeeping. All attendees are muted on entry. We'll be using the Q&A feature to collect questions related to the presentation. You can enter questions for the presenters as they occur to you throughout the webinar, and we'll plan to discuss as many as we can during the Q&A at the end of the webinar. I think we'll also be able to open it up to let people verbalize a bit too. If they want to come off mute and ask questions verbally, we can do that as well. Chat is currently visible to all, so please use it to talk amongst yourselves and ask organizers support or technical questions. And I believe one of the poll questions, if there's poll questions that have open answer, you can use the chat function for that as well. So on the next slide here, I'll introduce our speakers. Joining us today are co-leads Chris Peter and David Burdick. Chris is the research coordinator at the Great Bay National Estuarine Research Reserve. David is a research associate professor of coastal ecology and restoration at the University of New Hampshire. Also joining us are Grant McCown, a research scientist in the coastal habitat restoration team with the Jackson Estuarine Lab at University of New Hampshire, and Alice Yates, who is the stewardship coordinator at the South Slough National Estuarine Research Reserve. And also, and I want to leave Lauren out. Lauren White is also a scientist, a biologist at Great Bay Reserve, and she's going to be handling a lot of facilitation and back end stuff. So you'll see more of her from me at the end of the webinar. And with that, I think it's up to you, Chris. So go ahead and take over.
Great, thanks, Nick, and thanks for helping with playing this webinar and providing this platform for us to get our great research out there and other people's great research. I also want to thank Neuroscience Collaborative in general for funding this project. Neuroscience Collaborative is the main funding arm of the National Estuarine Research Reserve System, and we find that very valuable to add to the coastal collaborative research community. And I also want to thank Dave, co-lead, everyone on the project. Kim is also the co-lead. She's not presenting, but her fingerprints are all through this. She's led so many different portions in terms of data analysis, visualizations that we'll get through. So many more people to thank. I do want to say, like, our speaker lineup is great. We have people from East Coast to West Coast with Southern accents to New England accents to Australian accents from 3 different generations. And so you have a great diverse set of voices. And we're also working on the backbone of the NERRs system, National Estuarine Research Reserve System. Over 20 reserves are involved. We're using all their data set from over a decade. We have partners from over 14 external agencies advising on this project. So it's a huge, huge team effort. So it's not just the speakers here, it's so many other people involved. Before I jump into what this project is about and how we did it, which is the typical way, I'm gonna actually jump us into a poll. I almost forgot about that. But if you haven't already, please take out your phone and take out your phone app and scan this, and it'll bring you right to the website. Or you can use that link in the chat or right here. If you type this in, it's not quite a link. And then I'll pull up the actual poll because we want to understand a little bit more about the audience. What brings you here today? And again, here's the QR code here on the bottom left if you need to jump on and use it and start answering. And you kind of see real time what brings the audience people here today. Is it piles of, oh, wow, that's quick. I can't keep up with that. But we have, all right, we have like 33 people responding already. That's a good response rate. A lot of people want to understand natural trends. Some people I want to be informed of marsh management. I can't monitor the chat right now, or can I? Oh, I can, but nothing there quite yet. But I'll give you another like 15 seconds or so, just to get a little bit more response rate here for people that are a little bit slow with their phone or their internet web browser. All right, gonna move on to the next question. Yeah, I think I can just advance it. Yeah, so how have you contributed to the study? And this is kind of contributed is like in a loose way. Have you collected field data? Are you involved in any other meetings? Are you new to the project and just really here to learn about it? So please see, please respond, whichever makes sense to you. And I think you can select more than one thing that applies. Great, we have a lot of colors here that are kind of similar, but it looks like most people are kind of new to the project and look forward to hearing about it, which is great. Seeing a lot of people have attended a meeting before, followed by a lot of people who collected field data, and we're really thankful for that. All right, up to 50 responses out of 85, looking good. Give it another like 15 more seconds. Stats are holding true. This pastel donut chart looks really good. All right, I'm going to transition us out and back to the slideshow. Before I get into, I was just saying, before I get into like what this project is about and why, I wanted to give a little bit of elevator pitch about this project. So I didn't put a title in here, so it's just Namaste, and I'm hoping you understand what that means. But a lot of people don't. A lot of people are new to this project. I'm trying to wrap this up in one sentence, and it really means I've had 4 years to figure this out, but it really means data, and more data. So we're standardizing it, we're analyzing it, we're visualizing it. And when I say it, we're doing it for tidal marsh plant community change in response to sea level rise and climate change across local, like slight reserve level scales to national scales. So that's the kind of elevator pitch for this project. And now I kind of want to jump into why we did this project, which is a little bit different. than what normally you start off kind of presenting on a webinar, but I kind of think it might be important. So we're in this marsh in Great Bay, New Hampshire, on the coast, and just pretend you're on like really tall stilts and you can move really fast. And what are you seeing in the marsh that looks like signs of degradation? So here in the low marsh, you're running really quickly across the mud flat. You can see some areas of dieback, some like calving, some erosive forces. You sprint all the way to the high marsh here. You see some lush areas, and then you see some areas that are not so lush. Hummocky, lots of ponded water, less vegetation than you probably expect to see. Take one last sprint to the upland edge where the boardwalk is. You're seeing dying trees, dead trees, shrub communities come in, some invasive species here. So these are the changes we're seeing. And you can get a better, more traditional bird's eye view of it. You can see a lot of water ponded here. You can see some dead trees at the edge of the forest and islands. And so this is kind of the story of change we're seeing in Great Bay, New Hampshire. We're capturing those stories across the country, redocumenting it in kind of like these video and picture and like these short story forms throughout. That'll be coming later. And I'm not going to replay this again, but I want to zoom us now into the ground level where we're actually doing the monitoring. So we've been capturing these changes for over a decade using a national standardized protocol across the reserve system that looks at the effects of sea level rise climate change in marshes. It focuses on plant communities. We're also looking at tides, elevation through surface elevation tables, and RTK and other surveying techniques, and faunal communities as well. But for this talk, we're going to focus on the plant community. So this is the low marsh right in that drone video that I just showed, and you can kind of see like a lot of vascular plants. The Spartina ultimiflora here in New England would occupy this marsh is being replaced by macroalgae. So some fucoids, some alva, and some bare spots moving in. If you go to the high marsh, here's some of my colleagues in very firm footing. This is peat, but it no longer can sustain vascular vegetated marsh plants because of excess flooding and lack of drainage from historical embankments from farming practices. And lastly, here's a really confused me from this summer that's standing at the upland edge of a marsh next to a dead tree, dying live tree, and salt marsh like full halophytic plants of Disticulus and Spartina patens, which is salt hay or seaside goldenrod, and about a half foot of seawater. So things are certainly changing. We're noticing those changes. Now I'm gonna do one more video that's hopefully won't make you too dizzy, but if you follow my cursor here, this is the flight path of the drone, and this is across a series of monitoring plots across a transect. And then I'm just gonna stop it here just because I'm gonna be talking too slow compared to my video speed, but these are plots, these individual yellow points, just to orientate you to our data in our protocol. These are transects, and they run from the bay edge to the forest. And this is very common for all the reserves and the sites across the system. We monitor annually around peak biomass. There's 3 to 6 transects typically. And this is one site in Great Bay out of multiple. There's one on the East Coast. There's one on the northern shore as well. We created this tool here to help us understand and visualize the data set that we're dealing with, because we've never been to all these sites as well. So we as a national synthesis team are trying to understand these sites better. So we built this tool. It scales, it zooms out to the entire nation, even including Alaska. We have over 20 reserves, 80 sites, 20,000 plots of data. There's different ways you can visualize it through microtidal, mesotidal, just different tidal regimes, different salinity regimes, different samples, years sampled. This is looking at distribution. So we have just different ways to visualize it. We also hope to add some results here through the expertise of Susan Shells on the team and create this dashboard. So just some things that we've been moving forward with. Now I kind of want to get back into the what and how. So there's a little bit of a detour, but so what is this project about? It's really just to better understand tidal marsh change that informs management. That's the high level goal. So how do we do this? I'm going to try to tackle in one really quick slide here because I really want to get us into the results. So we did it in 3 phases. I'm going to call this the salmon phase, which is probably the hardest phase here. And it's about data collection and cataloging, which I just showed you, and also wrangling. Lots of wrangling. I don't have any slides on that, and you probably thank me later for that, but that was probably the bulk of the project. And now I'm going to call this like the seafoam green phase, is the meat and tofu of this project, which is the fun part. We really analyzed this huge data set at two different scales: the reserve site level, which I'll use interchangeably because you can analyze at a reserve like a collection of sites that are nearby or just one site. So it can be interchangeable for people that don't have sites within the reserve system, and at the national level. And then lastly, at this blue phase here, we're looking at communications like webinars. We had a training webinar, we had in-person workshops. We're going to have a great communication platform for their website and some video and some illustrations, and you'll see that throughout. So that was the process. Now I want you to pause for a second. Now I'm going to talk about reserve level results, and I'm going to go through one last video here, and this is really just going to showcase you what type of visualization analytical tools that we created that could be useful within the reserve system and outside the reserve system. And this was done through our studio, done by our great statistician, Kim Cressman. And so one of the components here is data exploration. So you can kind of see what sites you have, how much data you have distributed across sites, how does it look spatially. You can look at a time series of data for different specific categories. We have a new metric called ecotone migration index, which I'll talk about later, which is a really exciting new way of looking at marsh change. These bar charts are very customizable by vegetation zone, by site, by spatial bar charts. If we want to really hone in at the plot level, look at change. Again, this will be way faster than I can talk. We also have univariate analyses here, linear mixed effects models, looking at time across zone and the interaction, using post hoc tests. Sorry, your brain's probably spinning here, trying to keep up. We do a lot of multivariate community ordination. I love ordination, so I'm going to show this way too long, but here's a two-dimensional graph. Here's a three-dimensional one, interactive one that kind of just shows differences in plant communities across zones, across time, and what the factors are contributing to it. And then one last ordination, I'll get off of it, is here's a way to like use the year as a centroid and seeing patterns temporally kind of easily. You kind of see that this trend, and this is in Narragansett, Rhode Island, it kind of shoots downward, so you can see that change. And then how does that, do we know if the change is significant? We can run tests like Permanova, which is analogous to ANOVAs. And I'm running out of breath here, but then you can understand, using a simpler test, understand what is causing these changes or what are driving these. And for instance, I was showing you high marsh in that time series, and Peyton's here is gone from 70% basically to 25% driving that change. And that's a lot, but that was just kind of a flashpoint of what type of visualization tools we have. Now, lastly, I wanted to kind of point you towards reserve level results. So this time I want you to pay attention to results. I'm going to highlight different coasts here. And feel free to facilitate me, Nick or Lauren, if I'm running out of time here. So I'll try to do this quickly. But on the West Coast here in Southern California, this is species richness on the y-axis and time on the x. You can see that there's a decline in all four zones here, denoted by different colors. And this is in California. If you zoom all the way up to Alaska, big coast, you can see a difference, like very opposite pattern. This is Shannon-Wiener diversity, which is similar richness, and we can see that increasing over time. So why? Well, first of all, it's hugely different geography. The second, we have sea level rise of about 5 millimeters a year in Southern California, and then we have around -8 in Alaska. And the ocean is rising in Alaska, but the continental rebound is rising faster. So that could be part of these different responses. But we also see some different responses, mixed responses in Oregon and mid-coast California too, on the West Coast. Now jumping into the Gulf of Mexico, we can see, kind of want to highlight this different response in Grand Bay, Mississippi. And this is a spatial bar chart. So these are all plots like spatially across a lat long. And you can kind of see some patterns. And a couple of real quick patterns here are spartine ultrafluor and these lower elevation zones across these three transects are decreasing over time. As you can see my cursor, I think you can see that. And then Juncus, the other dominant species, it's a bit more upper elevations. They're also decreasing over time across each transect for the most part. And we have these kind of silly but fun icons to help you steer your mind throughout the presentation. So just want to make sure I kind of identify that. And lastly, I want to say with this, the Gulf of Mexico is changing the fastest of all the regions we looked at around the country. Lastly, I'm going to bring it back to New England, to New Hampshire, and just point out this ecotone migration index. And before I interpret these results, I want to jump into what that means. And so we've been calling it Ecotone Migration Index or EMI or EMI for fun, using a fun, silly icon here. And basically, after you get through the fact that this is an EMI word holding our logo, you can try to understand what this is. So basically, we're asked the question, with greater sea level rise, we're expecting these changes in these zones and these sites. And so, for instance, in New Hampshire, in the high marsh, with greater rise of water ocean levels, we're expecting to see greater covers of bare water and sparse flora, and we're expect decreases in the cover of other halophytic vegetation and brackish vegetation. So that's simply it. It helps capture responses that are species specific, that vary across zone and vary across bioregion, and it helps us compare across the country too. So getting back into New Hampshire and the results, here's EMI on the, or ME on the y-axis, time on the x. These are 4 different zones, and they're all increasing. And that is all signs of sea level rise. I did also want to mention that New England is increasing at the second fastest, or not increasing, I would say. It's showing the greatest signs of change, second most across the system. Lastly, before I hand it over to Grant and Alice, I did want to show that I've showed 4 marshes across the country, but there's over 80 marshes that we looked at this reserve level, site level. And how does this compare? Well, I compiled all the tests into one, like almost like a meta analysis. And so this is the compilation of all the tests. We ran about 7 univariate tests for every reserve. And I just wanted to see how often did we get a significant univariate test? Was our p-value lower than 0.05? And for most of the time, we're above all the time, we're above 60%. So lots of change throughout the system, mostly in the Gulf of Mexico. And then lastly, I would say I did the same thing with community ordination, and we also found a lot of change compiled. And I think, hopefully I'm doing well on time here, but I'm going to transition it over to Grant to talk, and Alice to talk about national level results.
All right, thanks, Chris. So I'm going to open us up to transitioning from our reserve analysis. So we did each of these individual site reserve analyses that Chris just talked about, and we compiled them all into a large data set consisting of 56 marshes and over 15,000 plots to look at general national trends. And the goals of our analysis were the what, the where, and the why. So first, we wanted to understand what vegetation metrics were changing over time. So just like in our reserve analyses, we use certain vegetation metrics that might show change or might be perceptible to change, such as halophyte cover or abiotic cover or that IMI or even species richness. We also were concerned about where are the changes in the vegetation community happening? Are they happening in different regions? Are they happening in different salinity regimes or tidal ranges or geomorphology? And lastly, we want to know why is the vegetation community changing? So we looked at certain metrics that were already calculated or monitored, including sea level rise, maybe the surrounding landscape condition, or how degraded the platform might be. And so we applied a series of screening guidelines to filter or subset out that national data frame from all the reserves that were compatible and similar to each other. So we used a minimum monitoring length, a minimum number of plots per site, and the habitat had to be a halophyte-dominated salt marsh that hadn't undergone recent restoration. So with that, from our reserve analysis, we had 20 reserves in 79 sites, and after filtering, we had 20 reserves in 56 sites. And just to like highlight what kind of sites got removed, we removed 4 sites because they didn't have enough plots per site. We ended up removing 5 sites because they didn't have a minimum number of monitoring seasons within 5 years. And we also removed a fair amount of sites because they weren't within our habitat guidelines. So we used 3 statistical approaches to conduct this national analysis. First, we used univariate analysis, which consisted of both national linear regressions and a compilation of site level regressions, which is super similar to what Chris just showed to answer what vegetation metrics are changing. We use multivariate analysis or ordinations like what Chris also showed to kind of really understand how might our marshes be changing. And lastly, we used model selection and linear mix modeling to understand why our model's changing. And I'm going to talk about our univariate analysis. And Alice is really going to talk about our multivariate and our linear mix modeling. And so through our national analysis, we found 5 major trends where really almost all three statistical approaches supported them. First, that our marshes are changing and they're changing at different scales, including at the site, the region, and the national level. Second, we found that sea level rise was the main factor across for this marsh change. Third, we found that how the vegetation community changed was impacted by the salinity regime it was in. Fourth, we found that marsh change was also impacted on which zone you were in. And for background, we divided all of our marshes into different zones, into 3 zones. They were low, high, or up, or low, mid, and up. And lastly, our 5th trend was that marshes are changing based on what region they're in. And like Chris highlighted, it seemed that the Northeast and the Gulf of Mexico changed the most. So I'm going to talk about the first trend. And so for each trend, we have a title screen like this. And for reference, we have shown which statistical approach that we're going to talk about in this. And just to highlight, almost every trend was supported by all 3 statistical approaches. And for the first trend, I'm going to talk about the univariate analysis. And so just a quick background of that univariate analysis, we approached it at 3 different geospatial levels: at the site level, at the region level, and at the national level. We use 3 different approaches to try and understand how or which vegetation metrics are changing. This is their first approach was using site level regressions, which is what Chris kind of showed earlier. So we would run a linear regression for different vegetation metrics. For example, this is one site at Grand Bay. We then would scale that up to all 56 sites across our study. And then with these calculated trends, we could take the average and the standard error, let's say, of our slopes of live cover in for a certain site characteristics. So for example, this top graph shows the average slope for different tidal ranges. Or we could do what Chris showed earlier and calculate the number of sites with a significant trend, like I did with Shannon Wiener diversity in the salinity regime. Our second approach was we were really interested to see how vegetation communities changes with respect to site characteristics. So we did some national time models with using site characteristic as an interaction. And as an example, if we want to look at 2 salinity regimes where orange was oligohaline and blue was polyhaline, we were really curious if there was a significant interaction over time. And lastly, we were really curious just about national general trends as well. So we used the same approach of a national model, but with no interactive effect. So for example, here are the same points, but we would just do one linear regression. And so for the results for the first trend, I'm just going to highlight a few vegetation metrics. So at the site level, showing live cover, we found that over 50% of our sites had a significant or a near significant trend. So the green is regressions that were significant or a p-value less than 0.05, and the grays are those that were trending significant or at least a p-value less than 0.2. When it came to our famous EMI metric, we also found that 50% of our sites had a significant or a near significant trend as well. Looking at the region level, and I'm going to talk about this more in depth later on in the talk, but I just want to highlight this graph where the y-axis is visual cover and the x-axis is time, where each color is a different vegetation metric, just to kind of show how different ones are changing. And I just really want to highlight that the Northeast and the Gulf Coast are losing a substantial amount of live cover, which is in the red, and halophyte cover in the green. I also just want to highlight that the Mid-Atlantic is actually gaining vegetation cover and in respect to live and halophyte cover. And then a few results at our national level. First, to show halophyte cover over time, we saw on a national level that it was losing about we were generally losing 0.5% halophyte cover on the national level. And when we wanted to look at our EMI, we actually saw increasing EMI on the national level as well, showing that our vegetation is shifting into those upper zones. And so next, I'm going to give it to Alice to talk about our next 3 trends.
Thanks. And Grant's going to advance my slides. So So the next trend is sea level rise is a main factor associated with marsh change. And I'm going to talk about our linear mixed effects models next. And so essentially we built these 4 linear mixed effects models where down in the bottom you see the pink boxes. So these are the different responses that we looked at with linear mixed effects modeling. So we created a model for each one of these response variables, and then the boxes in the orange are explanatory variables that we included in our full model, allowing for an interaction with sea level rise and proportion low marsh, which is how we dealt with zones at this scale. And then we compared all of the subsets of these models using AIC, which is Akeiki's information criteria, which is a way of looking at what model best fits the data. Next. So we're talking about sea level rise in this trend. And so for 3 out of the 4 models that we created, you can see in the boxes down below, had sea level rise as a main explanatory variable in the best model selected. And I forgot to mention, we do have reserve as a random effect. Next. So here's what that looks like. So on the x-axis down below, it's rate of sea level rise of indicated where sea level rise is decreasing and then increasing. And so what you can see on the left-hand side is our ecotone migration index. So this is the indicator of cover class changes that we would expect. You can see that we have a positive trend, so these are increasing over time. And then on the right-hand side, you can see changes in live cover. And you can see that there's a interaction effect here with geomorphology, but generally each of these 3 types of geomorphology are decreasing in percent live cover with increasing sea level rise. Next. So trend number 3 is marsh change differs across salinity. Next. So I'm going to stick with the linear mixed effects models here, and you can see that salinity category is in all 4 of the models that we assess. So that's the best model for each of these response variables had salinity in it, either as a main effect or as an interaction term. Next. So here's one example from that. We've got change in live cover on the y-axis here. And this is proportion low marsh. So next. So you can see here, we've got marsh on the left-hand side is a minor component, low marsh. And then on the right-hand side, low marsh is a major component. In the orange line here, this is where low marsh is minor, and then those least saline sites, the oligohaline marshes, are declining in live cover. Whereas in the blue there, low marsh dominated areas, polyhaline, so those higher salinity level marshes are declining in live cover. So you can see there's an interaction effect depending on how much of the marsh is low marsh or not. Next. So then trend number 4 is marsh change differs across zones. And we're going to talk about multivariate analysis here. So next. So we use non-metric multidimensional scaling. And so this shows dissimilarity between our plots. Sorry about all the text. I'm going to show you some visuals in a moment. But I wanted to note that conventionally this uses species cover and abundance. But the challenge with national data is that if we use species cover, this would basically just show the species ranges, which wasn't interesting for this study. And so we use cover classes to standardize, so halophytes, fresh, etc. And then we also looked at rate of change per cover class. So each of the points that you'll see is a change rather than an individual percent cover. Next. And then we use BioEnv, which is a way of assessing correlated environmental variables such as sea level rise, zone, region, etc. And other categories such as diversity. And that overlays these axes on our NMDS plots. So next slide, I'll show you what that means. So just to walk you through it, so this is all of the data across the nation. Oops, what's going on? Okay. So I'm just going to show a few slides to orient you to this plot. So where the box is there and the arrows, these points have very different rates of change per cover class because they're very far away. So points that are close together have a very similar rate of change on those cover classes. Next. So this points in this direction show the greater change in diversity, so increases in diversity. Next. Points in this direction indicates marshes keeping up with sea level rise and increasing or little decrease in percent live cover of plots. Next. This direction indicates greater rates of sea level rise, increases in unvegetated cover, and increases in ecotone migration index. Next. So we're going to look at one region just to actually see what those points are doing. And so this is the Gulf Coast. Next. So what you can see here are these squares. This is the upland edge trends are more similar to each other than other zones. So you can see they're clustered over here on the lower left and are increasing in diversity and are less correlated with sea level rise, which runs along the perpendicular axis to this direction. And so what we're expecting here is when trees are dying off, you're actually getting an increase in diversity because those understory or those ground cover species are encroaching in here. Next. What we see here is low marsh trends strongly with sea level rise, as well as live unvegetated categories. Next. So just to sort of point out that, so on the top of the screen there, marshes are keeping up with sea level rise, total vegetation not declining, ecotone migration index values are good or better, and then down on the lower right, rate of sea level rise is high, becoming more unvegetated, and ecotone migration index values are high. So this is the poorer end of the spectrum. Next. And so I just wanted to point out that including zone as an interaction effect in models, it increases the likelihood of detecting change in marshes and increases our understanding of where marshes are experiencing the most change. Because if we just looked at it without an interaction effect, sometimes different directions of effect that you're seeing in different zones could cancel each other out, and it would look like nothing is happening at the whole site level. Next. Back to Grant.
Awesome. Thanks, Alice. So I'm going to hit on the last trend in our national analysis, which is looking at how marshes change is actually different across different regions. And I kind of highlighted it earlier, but I wanted to go into a little bit more deeper, show a few more graphs. And again, we're going to be using a univariate analysis. So again, I wanted to highlight this graph again, where I've overlaid live cover in red, halophyte cover in green, and freshwater cover in blue over time for each region. And I really just want to highlight again, I think this graph just tells so much of the picture, is that the Northeast and the Gulf Coast are changing the most, and they're losing vegetation cover over time, whereas the Southeast and the West Coast are generally not changing over time. Whereas the Mid-Atlantic is actually gaining live cover and halophyte cover and freshwater cover, interestingly. Additionally, we also conducted site level regressions, or looking at a compilation of the site level regressions. We compiled them in a different way for IMI because of its just because of the unidirectional understanding of it. So the way to read this graph is that if a site regression, if a region has a score closer to 2, it means most of its sites had a significant trend and that slope was positive. Whereas those, if it was closer to -2, would have also significant trends, but would actually be negative trend for IMI. Whereas those that are close to 0, such as the Mid-Atlantic, the Southeast, and the West Coast, meant that the average of their sites really were that most of their sites did not have a significant trend overall. And so what I just wanted to highlight here is that the Northeast and the Gulf Coast had compiled IMI scores of greater than 1 on average, meaning that they have more sites that have significant trends and that the ME score is positive. So just another way to view a region level view of ME. Additionally, I also want to highlight species diversity as well. I found this result really interesting, where if you're looking at Shannon Wiener diversity over time, we saw that the Northeast and the Gulf Coast also, on top of losing vegetation cover, were also losing species diversity in general across their sites. Where again, as the Mid-Atlantic was quite stable, and the West Coast and the Southeast actually saw gains in their species diversity. So if there's anything you take away from this site or from this talk about our national analysis, I really just want to hit on all 5 trends again. That first, that our marshes are changing, and they're changing at all 3 scales. They're changing at the site level, the region level, and the national level. And we found out that sea level rise is the main factor associated with this marsh change and is causing the change in vegetation community. Additionally, there were certain factors that would impact how vegetations would change. In trend 3, we saw that with the salinity regime. In trend 4, we saw it across different zones between the low, mid, and high zones. And we also saw that in the at the region level too. If you're in the Northeast or the Gulf of Mexico, you usually saw declines in cover and species diversity, whereas in the Mid-Atlantic, you actually saw gains in cover and species diversity. And with that, I'm going to transition it over to Dave to talk about looking towards the future and some of the deliverables for this project.
Hey, thanks, Grant. Now that you've seen major results, what I'd like to do, and the takeaways with those, I'd like to do is step back and review some of the project impacts. So these Namaste tools that we've all developed, especially with help of Kim Cressman, can be used over time. And you can use them at different sites if you wanted to add a site to reserve, or if you want to include maybe a state group really loves your results and they want to start setting up a site and including that. It can be set up for that. And you can even do different ecosystems. And so as we expand our examination of ecosystem health in our reserves, you might want to include upland areas, fields, seagrass beds, things like that. The ME metric is a unique way to track sea level rise effects. Essentially, it's based on what we expect to happen with percent cover due to sea level rise. So we're all out there measuring percent cover on these transects for whatever's there, vegetation obviously, but rack and dead plants and bare soil. So by suggesting which ones may change ahead of time, we've kind of been able to set up a cool tool, this ME metric, a cool tool to look at sea level rise effects. We based this on a Spartina-Alterniflora ratio in New England because most every marsh had Alterniflora in the salt marsh. We could do that, but we couldn't do that when we went across the country because there's many marshes that don't have patents and even Alterniflora, especially on the West Coast. It's an invasive. So by using this ME, we can transcend that limitation we had from species and just talk about what species we sort of expect to get kicked out as sea level starts rising. So that's super cool. And the other thing I wanted to bring up is the NERR national protocols. We've been using these protocols now for 15 years. And our project shows that you just can't work with data that aren't compatible. So some of the data collections were pretty spot on and collected it, the right way. And we've been able to deal with 2 major groups that say, no, I want to look at visual cover versus point intercept cover. But one of the things we haven't been able to get around is, plant vigor. And so the height of the plant relative to its normal height or the highest height and how vigorous that plant is a real problem when you have some people collecting data from 4 corners of the plot, some people collecting from the whole plot, some people collecting for 4/5 to get a canopy height. So if we could standardize that would really be helpful. We could use that data, but we can't the way it is. Next slide. And I also wanted to talk about comparison with other national tools. The NERR has done just a fabulous job. The teams, you guys are really the best and leading the nation in developing these large-scale models to understand how changes are occurring in our marshes. We've had Mars with Kenny Raposa, and all these are illustrated on the right. We have a landscape resiliency from Rachel Stevens and her team. We have UVVR from Neil Ganju, that the images from Grant's work in one of our sites. But that's another way to look at marsh vulnerability and impacts. And finally, there's our Charlie Andrus's brand new publication from the High Tider Project that looks at how marshes have in the estuaries, but vegetated wetlands at the edge of the estuaries really have changed over the years because of all the things associated with human impacts to the land. So I wanted to just call your attention. So what Namaste does is provides more of a call of action to stop marsh loss without saying exactly how the marsh is being lost, just that we're measuring this marsh and we're seeing changes. The ME is increasing, so we're losing, losing marsh health. And so but it does provide clues on causes and actions. And so this kind of leads to the next phase is what is your system or your region really thinking about in terms of marsh restoration or restoring or preserving marshes or increasing the resilience from sediment deposition, freeing up sediment so that there's more sediment in the water column, nature-based solutions like living shorelines. I work a lot on hydrology, fixing channel and surface hydrology issues, and marsh migration's really big and important. It's been dealt with by a lot of the NERR national tools. And this High Tider kit, which is of course brand new, provides a great historical context and insight for those migration paths, as does Stevens. So you've done a great job with that. Next slide. So the next steps, well, we have this dashboard, which I hope you go visit. It shows the whole country. You can zoom in, and that's what it looks like when you zoom in, Coggleshell Marsh, with the data sets kind of shown with different species changing over time. So this is a great product, but we also have other site level data tools, and you can go both online and you can create new analyses yourself using the R tools. In the future, we kind of want to work with different NERRs to refine the EMI where there may be opportunities to improve it, and also meta-analysis, so we can kind of look at the individual analyses for the nation, see how they're doing and how they can be combined, sort of like those slope regressions that Grant was showing. And finally, I want to talk a little bit about communications. We have the grant's just over, and so we have a lot of communications to do. We're developing some really cool illustrations. We have a brand new website, which is just a kind of a welcoming, very high level website. And you can get to that from the QR code at the bottom. Next slide. And this is just a sort of a treat for everybody. This is one of the communication products we're working on right now, and it shows from left to right the changes in salt marshes we're seeing in the data sets from different sites and how those may be seen as, through over time from left to right and in 3 dimensions with sort of things that we don't collect in the marsh, like animals and houses and where people live. But still, I think it's gonna be really neat. I hope you like it. Taylor Rose is doing these and through Roker Communications. And I've just seen the next version of this morning, and it's really good. So there's some things that make this better, and I think they're being made better. So I'm pretty excited about some of these communication products that will be coming out over the next 3 or 4 months for this project. Okay, next slide. Okay, I'm going to hand this back to Lauren, and she's going to run a poll for you.
Thanks, Dave. I'm going to take over for this one just to not make people's heads spin too much with different speakers. Did want to just make a good big shout out, emphasize Dave's shout out about Taylor Rose and Roca Communication. They built the website for us. Roca, Dolores, and Lola did in like 2 days. So it's really just for here, and it's in draft, but it houses a lot of our products and our future products. So please visit that. I think we have a slide with that, or it will be in the chat for our website. But before we jump into that, please go back into this this poll here with the QR code. And Grant, I'm going to take over sharing so I can share the actual Mentimeter again, if you don't mind. Thanks. And hopefully I can get this right. Maybe Dave, you can give me a thumbs up or thumbs down if you're seeing this. Yep, great. So now that you've seen this great presentation from great speakers, what are the main project elements you find most valuable? Let me move this thing out of the way here so you can see all the questions or the results. Is it site level? Data tools, reservable results, EMI metrics. I am reading them in a very weird way. Lots of likes. A lot of people are really into the results. No big surprise. That's where I like to go. And I just want to see. Oh yeah, site level tools as well. Now I get 44 responses. I'll give another like 15 seconds. All right, get your last final responses in. And the one last question. I know we've taken up a lot of your time here, and time is valuable. There we go. Last question. What would you like to see in the future? And so you can select up to 3. So use your likes carefully. Is it a national story map? Is it greater regional analyses? Is it data tools for new users? Is it something that we don't have listed here at all? Is it 2-page reports that are more for the general audience? Is it live supportive data tools? So we get a lot of cool animations here coming in. Just give you 20 seconds to fill this out. Yeah, a lot of buckets here. Yeah, we might have to work on this for longer. Yeah, data tools, national story map, greater regional analysis. Yep. I want to see all these things too. This is great. If you have anything else that comes to mind, please write it in the chat or feel free to respond. I think we might have a poll that Nick might send out. Maybe yes, no. Nick, but tell me if I'm wrong. And if not, you can always email me. I think my contact information is on the website.
There is an exit survey that you can use to drop in additional questions on the way out of the webinar.
Great. Thanks for confirming that. Give this another like 20 seconds to get your last few thoughts in. Feel free to write something in the chat too. And then I think right now we're gonna just turn it over to Q&A. So if you have any questions, write them in the chat. I think we're gonna come off of screen sharing and allow attendees to really turn on their cameras, and so maybe all the speakers can come off, put their cameras back on. Yeah, if anyone has any questions, feel free to write in the chat, and then I think Lauren's gonna facilitate us on the question.
Yeah, and Kim has just put some valuable information in the chat about data tool training. And she put a link for the data tool training that exists right now. And
yep, so everyone that voted data tool training, check out that great resource. And if there's some additional things there that you're looking for, please let us know. But it's pretty impressive what Kim put together.
Great. Well, thanks for those presentations. I've got a couple questions that came in through the Q&A box, so I'll verbalize them here, and then if no more questions come in, maybe Nick can enable people to come off mute. First question I have, how did you define or determine low marsh versus mid and transitional marsh?
Oof, I guess I should take this one on, but I'm happy to try to start and then let other panelists kind of answer the question. So yeah, we thought it would be best to leave it up to local experts. So we give guidance on local experts on how to do that. So in our mind, marsh zone was a expression of the plant community, which assimilates the physical conditions and biological conditions best. So I'm going to try to make this a little bit shorter, but we basically focus on either plant communities. So for instance, in the Northeast or entire East Coast, spartina alterniflora would be the determining factor for tall form alterniflora. And so that was one way. Another way is looking at elevations, looking at elevation distribution across the marsh. Another way is inundation, so water levels. And so there are some tricks to that because you're not always like elevation and plants are not always tied, but most of the time they are. So we basically the way to summarize it is we asked local experts to categorize it based off best available information from that guidance. Does anyone else want to add to that or make that more clear?
Yeah, one of the things is geomorphology. So, flooding and elevation and the plant community all depend on the geomorphology of the marsh. And in New England, we often have a steep, low marsh that grades into a high marsh. But the high marsh is relatively flat. And so it's that slope break is pretty easy to see.
Great. Thanks for the question.
Yeah, great. Thank you. All right. Question number 2. Do you think that one could predict the differing effects of sea level rise along the West Coast just by how much uplift there is, or might there be other factors as well? If the latter, what factors would you most recommend considering? Maybe Alice, our West Coast rep?
So we did look at surface elevation change with our SETs in relation to sea level rise. So factoring in, I know that uplift is kind of a big thing that maybe is not always captured with those. But it did seem like that was a pretty important driving factor, was the relative marsh surface change relative to sea level rise. We did also have geomorphology come into effect with our models at the national level, and salinity regime. So we were seeing different effects in different parts of people's estuaries in terms of salinity. So I think we were pretty limited in what we could look at with other, driving factors. We did include condition, landscape condition, and that's a larger topic and metric, and found that it wasn't coming up as being important in our models. So yeah, I don't know if anyone else has something to add to that, but.
Yeah, that's a great question and a tough one to answer. I would definitely just building off what you said, Alice, because I completely agree. Is we looked at, I guess the expression of that would be like SCT, surface elevation tables, and sea level rise, and what is the net effect of water levels. And we did look at that, but we didn't have a complete data set for all of our sites, and so we had to remove. We couldn't look at it for all our sites with that in the model. So there was a little bit of a limitation there. We had SCTs for, I don't know, I think like 3/4 of our sites we had that data, so we were able to look at it for a subset of it, but not all of them. And I would kind of guess that uplift is a big part of it.
Great. We're coming up against 3 o'clock, and I only have one more written question here. It is, were you comparing the same plot to the same plot over time? For example, a plot as a random factor in the model. Maybe again, Alice or anyone?
I know in univariate, when we did site level regressions, we did not use plot as a random factor for that, for those models. For the national regressions, we did use site as a random factor to account for that. Yes.
So, and at the national level, we looked actually at trends over time. And so those were at the plot level trends. So the trends were calculated, I believe, for plot level and then averaged for the site level or something like that. Kim did that fantastic data section.
Great. Okay, another question came in. Thank you. It says, did you notice any instances of breakpoints or drastic changes in your responses that might have driven or deviated from the overall trend?
I think we just looked at linear trends and didn't do breakpoint analyses. Is that correct, guys? That wasn't really in my
Yeah,
section.
that's the next level, really. There's a lot of data here, and there's a lot of opportunities to do work. And if somebody wants to look at those breakpoints, that's super.
Yeah, I think we did discuss it for site level possibilities, but it was pretty tricky to do at the, with so much data,
Well, one of the things we wanted to do also for everybody is we wanted to make sure that the seasonal variability due to climate wasn't going to overwhelm our trends over time, our 10-year trend period. And so just recognizing that that was true, the seasonal variability did not overwhelm the signal was really important to us. And so we were very happy, and we didn't want to dive in too much and see what sort of breakpoints we could find.
Yeah, this is a great question. I remember talking about in great detail about 2 years ago, like trying to see the pros of using a linear versus a non-linear, and using slopes to the national level versus a time series. And oh, I wish I could go back in time in my brain and figure it out. But there are pros and cons to each, and we ended up going with the simpler approach. But I think there's definitely the level we had a couple different plans for the reserve level and national level. We called them, what do we call them? Like a cheese pizza versus supreme pizza, and we ended up realizing we were trying to tackle way too much stuff. So we went to my chagrin, because I love everything all the time. We went to more of a cheese approach, but there's definitely some additional time to add some additional toppings.
Great, thanks for those responses. I've got another question I can read out here, but also if you would like to ask your own question, like Nick said, you can raise your hand and we'll try to get you off mute. The one that came into the Q&A box is, do you have capacity or plans to add new data to these analyses over time? So kind of that future impacts, future goals.
That's a great question. I think that's from, I saw it's from, yeah, Chris Janusik. I'm sorry if I butchered your name. I got your first name right, I think. We met and talked about this a little bit too. And I think we're taking a pause for now, but we are thinking about moving forward some other ideas to build off of it. As you remember, there's a potential national effort to do something similar with seagrass data around the country too, using a similar model of what we kind of mapped out here for Namaste and Tidal Marshes. I can't remember the directions we're really going at, but there's some awesome ways to move forward on this. Some of the ways that Dave mentioned was refine the EMI and conducting a meta-analysis. I know we've talked to Zach a little bit about that and Alice about that. So is there anything else that people kind of remember, that directions that we're thinking about, that brainstorm? Because there's a lot of different ways to go for sure.
I think one thing to note is that if you can wrangle your own data into the R code format and data format that we use, you can use the R code to analyze your own data. And so it won't be included in the national syntheses or regional syntheses, but you can get your own local products if you have this kind of data.
Yeah. Can we expand a little on the answer for Chris? Because, really, if we had really good elevation data, that could be valuable. And I don't think we had enough good elevation data everywhere. We certainly didn't have enough detailed salinity data. So there's definitely some things we could improve on over time and collect more information.
Just have to find funding for Kim to add that data in, and we can analyze it again.
Exactly.
All right, yeah, those are all the questions I've seen come in. So guess it's open. I think we had on our schedule an optional 15 minutes to go until 3:15. If anyone wants to linger and keep putting those questions in the chat or in the Q&A box, think we said we'd stick around for that time.
And I think that was because we all like talking nerdy about stats, and we figured we didn't have time to actually put in a lot of that detail into the presentation. So if you too like to talk nerdy about stats, you can answer questions.
Absolutely. Yep. If there's anything we can help answer with, or help use these data tools for, or are there any next steps you want to work on this with us, we have limited time, but lots of enthusiasm.
Hey everyone, can you hear me? This is Zach.
Yes.
Yep.
I would love to hear a little bit more about what your multivariate results were. You had a couple NMDS plots up there, but didn't get a full chance to look, and I'm curious about how that came out, especially not using traditional responses as for what you put in your matrix.
Yeah, Chris, did you want to talk about since you love it so much, you already said that you love it. So.
I do love it. I love it a lot. But I was gonna say I love it more at the site level because it's a little bit easier for my brain to wrangle because we're not using slopes. But there's all these different site level results, but the national results using slope. There's some take home messages. I'm not sure if Kim is still with us. I don't want to put her on the spot. But if she wants, you feel free to come off mute or mic, or feel free to just like put a bad emoji up if I get this wrong. I can just try starting it, but feel free to interrupt me at any point, Kim, because she's the one that conducted all the multivariate national level results. Okay, and so a couple of things that were noted was that some of the best predictors in the multivariate was sea level rise, the region, and the landscape resiliency of condition metric called condition. These were the three biggest ones that kind of were explaining our variables best, our multivariate changes. Some of the other things we found is that the best predictors at like a finer tune level, if we added in not just like a site level multivariate, but a plot level multivariate, using plot, zone, and site. We can see that plot and zone were huge predictors there. So these are things, especially zone, sorry. So these are things that were also just accounting for a lot of variability and a lot of the changes that we're seeing across the nation. I also want to repeat that region, which didn't quite make the cut in the linear mixed effects model. That was a big predictor here as well. So there's a lot of big things. I think they highlighted region and zone for sure. I'm probably rambling, so I'll probably just cut myself off. Feel free to jump in. Anyone else wants to?
One of the cool things is by the end of 2025, there'll be 3 more years of data, right? So as this data set extends in length, we will be able to see finer, hopefully, finer distinctions and maybe new and different effects over time as time goes on, as we get more data is added to this. So it's pretty exciting in that respect.
I had a follow-up technical question. Did you have to change your indices that you used in the matrix because you were using the slopes, or did you just stick with traditional Bray-Curtis? You said you wanted to talk stats.
I feel like I should come off mute for this, and Chris might breathe a sigh of relief if I do. So I used Euclidean distance in the NMDSs, and I believe I centered and standardized everything first. I would need to go back and check, but I sure hope I did that so that everything would have been on a scale from 0 to 1, and then Euclidean distance rather than Bray-Curtis.
Thank you. That makes sense.
Yep. Yeah. I'm looking at your nice summary, Kim, and exactly what you got it. You got your summary down 100%. Standardized and centered.
Thanks, everyone.
Thanks, Zach. Any other questions before we wrap things up? I know we're kind of whittling down our attendees list here.
There is in the chat rather than the question, Chris Janasek just mentioned, I would also like to suggest that in place of tallying significant p-values for some of your analyses, that you compile effect sizes instead, p-value. Yeah. So in terms of that, that's what a meta-analysis will do, is it takes the full kind of scope of replicate size, effect size, and some more information from those local site level analyses, and then compiles it in a formal meta-analysis which would look at trends across the nation and see how those site level analyses are scaling up. So that doesn't just use the p-values. So yes, we agree with you that that is a great next step, and that's what we're hoping to find some time to do, is run a formal meta-analysis that will look at that. If Chris is still online.
We got a thumbs up from Chris
Okay.
and me.
Okay, sounds like we're slowing down on questions. I'm happy to call it there. And if folks have any other questions that occur to them as we're wrapping up or on the way out, you can feel free to drop them into the exit survey. Thank you to everyone for joining us today. Thank you to our presenters for putting such a great presentation together and putting in all the background work that goes into making these webinars run. So it does take a lot of work. So great work to the team on doing most of that work for me, so I didn't have to do it, which is even better.
Well, you're doing the stuff we couldn't do, so thank you.
Well, thank you. Okay, so we'll call it there. And thank you to everyone. I hope you have a good rest of your day.
Excellent.
Thanks, everyone.
Great job, everybody.